This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode number 137, recorded on June 10th, 2011. Hi, everybody. I'm Vincent Racaniello, and this is TWiV, the podcast all about viruses. Joining me today from Western Massachusetts, Alan Dove.
Good to be here.
How you doing, Alan?
Oh, doing okay. Had some fun storms blow through the area, but today it's tolerable weather.
I heard lightning knocked out your router, huh?
Yeah, part of it. I can still get in on the Wi-Fi side, but it's a little bit glitchy. So if I suddenly vanish from the podcast, it's nothing personal.
Should we call you back if you drop out?
You can try, but I wouldn't be too optimistic. I may have to reboot everything from there.
Okay. Also joining us normally from North Central Florida is Rich Condit.
Hi, everybody.
And where are you today, Rich?
I am in Austin, Texas. I'm with my daughter and son-in-law and granddaughter, awaiting the arrival of a new grandson very soon.
Cool. Good luck with that.
It's all good. Yep,
yeah.
Indeed.
All right, have fun. Also joining me in my office, starting on my left, Dickson Despommier.
Hey, Vince.
How you doing, Dickson?
I'm doing well.
Were you in Korea?
I was.
And you built a vertical farm?
I didn't build it, but they did.
All right.
Really? For real? They built a vertical farm?
There is now the world's first vertical farm up and operational, run by the government of Korea.
You must have pictures, right?
They took so many pictures, I wouldn't know what to do with any of them. But it was the most amazing thing to see what was next to this vertical farm. It's the state-of-the-art, brand-new seed bank that was like 4 times as large as the vertical farm that they built, with high state-of-the-art DNA laboratories. Every seed species of plant in the entire southern peninsula of Korea is collected and stored there.
Are they contaminated with E. coli?
Not yet, but they're going to use the vertical farm to test the germination of the seeds. So it's a great new application for the idea that we didn't even think about something like that, but it's going to work out really well.
That's terrific.
But I'm glad to be back. I must say that I missed Tuesday, and I never found it. It's still out there, floating around somewhere.
It happens, Dickson, when you're an international traveler. Also joining us today, for 2 first-time visitors on TWiV, we have the John Snow Professor of Epidemiology in the Mailman School of Public Health, and also here at the College of Physicians and Surgeons at Columbia, Ian Lipkin. Welcome, Ian.
Delighted to be here, Vince.
Great to have you. And you're, I don't know what to call it, division.
My partner in crime.
Where you're from is the Center for Infection and Immunity. Is that
Many
a division?
Partners in crime.
Is that a division, a department, an empire? What would you call it?
It's a center.
It's a center.
It's a center.
Thank you. That's the word I was looking for. Center for Infection and Immunity, which I can see right out my window. I'm always looking at you guys.
He's always looking down on us.
It's only 2 floors. I like to see what's coming out of the chimneys.
Hopefully a lot of hot air and nothing else.
Not that kind of facility, we hope.
Yes, indeed.
Also joining us today, also from the Center for Infection and Immunity, the name of it's right in the title, Center, an assistant professor of clinical pathology, Amit Kapoor. Welcome, Amit.
Good to be here.
Thanks for coming. I met you a long time ago in
Barcelona.
Was it Barcelona, meeting?
Yeah, Sitges. It's a town near Barcelona.
That's right.
Yeah.
And you had, at the time you were working in India, is that correct?
No, I was in San Francisco with Eric Delbert.
With Eric Delbert, who we've also had here on TWiV. And you had just isolated these COSA viruses, right?
Yes, at that time, yeah.
Now, you got your PhD in India, right?
Yes, I was working on polio viruses, one of your favorite. And
I already like you.
That's good.
What part of India were you in?
I'm from North India,
Yes.
some place called Lucknow.
Oh, yes, very
Yes,
famous.
it's very rich in culture.
Absolutely.
So after you got your PhD in India, you did a postdoc with Eric.
Yes.
Was a postdoc, I
Yes,
guess.
I did.
And now you're here at the Center for Infection and Immunity.
Yes,
Okay.
I'm working with Ian.
And we're going to talk about your paper today, on a canine hepacivirus. In fact, that's why you guys are here. But before we do that, Ian, you're an MD, right?
Yes.
Where'd you get that MD?
In Chicago at Rush Medical College.
Rush, okay.
That was before I saw the light and moved over to basic science with Michael Oldstone.
So you had gone to medical school with the idea of being a physician, is that right?
I started off wanting to be a cultural anthropologist and realized that I had nothing to offer. That's a long story.
We'll do that next time.
We'll do that next time.
But then you finished medical school and you decided you didn't want to do that.
No, then I did internal medicine at the University of Washington, and then neurology at UCSF. And while I was there, HIV emerged. That was 81 to 84. I began working on HIV and realized that it took much too long to figure out what it was. It was causing gay-related immunodeficiency disease, it was then called. So Michael Oldstone took me under wing for 6 years. It was a life sentence, or felt that way. And then I emerged in 90 and went to work at UC Irvine, where I worked with Bert and Hung Fan and a number of other people well known to you. And then I came here in 2001, shortly after discovering West Nile virus, and just in time for 9/11.
Yeah, right. That's right.
Is this the last stop on your career?
I presume so.
Well, that would be
I'm
nice.
good.
Very good. All right. So we're going to have you back for another episode where we talk about all the things that you do. But today we're going to talk about this very interesting paper out of your lab, on which Amit is the first author and you're the last author. And
He's corresponding author, too.
he's also corresponding.
Right.
Let's see. If you
Thank
can
you.
find that here. Corresponding author. Here you go.
He's the dude.
At AK3117.
The
He's
big
guy.
kahuna.
And there are a number of people, very well-known people.
Thank you very much.
It's called Characterization of a Canine Homologue of Hepatitis C Virus. So I wanted to start out by asking you why, Amit, you decided to do this study. It says here you were characterizing the viral flora of companion animals. I guess that's dogs and cats. Anything else are companion animals?
Parrots.
Parrots.
They talk back.
Yeah. So
Medical students.
That's right. Ooh.
Presumably not aquarium fish, since we want respiratory viruses.
No. Monkeys?
So, Amit, why did you choose dogs?
So just after joining actually Ian's group, Ian was telling me about this interesting project from Pfizer that they were interested in doing, and I was just joining this, and Ian showed very interest in looking for viruses in companion animals, and then that's how it got started. And we were looking for, first we started some PCR to look for, like we were expecting picornaviruses, because it was very surprising for us to realize that there is no picornavirus of dogs known, and almost picornaviruses are there, so in every other mammalian species. So we started looking, then we tried to see if we can find, by doing 454 sequencing, something more than this. Picornaviruses and other common viruses, and that's how we came across this new virus that was similar to hepatitis C.
So you were looking in, you had already acquired samples from diseased dogs? Is this how this works?
So, yeah, we started, we talked with Pfizer group who was doing this, collecting these samples, and they were collecting these samples from shelter animals, because generally in shelters they are, these animals are ignored by their owners, and they are more susceptible to diseases because they are living in together from different places. And so we started there to expect that we will find whatever most interesting stuff is in these animals rather than looking in pet dogs. And then they sent us these samples from actually respiratory disease dogs, where they were not able to identify anything. So.
And Pfizer, this was Pfizer's veterinary branch, so...
It was their branch, but actually Ian guided them to pick those animals and ask them to, what kind of samples we are looking for this study.
So, Vince, they initially approached us to look at diseases of pigs and cattle and birds, things that would be economically important to them. And they were quite slow in getting us samples from the animals that they wanted to study. And I said to them, surely you must have something we can do. We can make lemonade. So they sent us these samples from the shelters, and Amit, grabbed the bull by the horns, trying to get back to cattle, as you can see, right? And ultimately found this very interesting new bug.
Well, you have a soft spot for dogs, right?
I love dogs.
There you go.
Right. So now this, initially you didn't know what you were going to find. You didn't set out to find any. You kind of had an interest in picornaviruses, but this was just an open-ended, what kinds of viruses are in these dogs, right?
Yes, but as I was involved in virus discovery before and after joining this group, I think anybody who is doing virus hunting, their dream is to find something related to hepatitis B, C, and HIV.
Right.
So means there was always an eye on any sequence that is close to these viruses. And then we found it, but we were not exactly expecting that we will find something like hepatitis C. This was amazing, yeah.
It also struck me, this is the kind of project that certain politicians might like to point to and say, gee, what a waste of taxpayer money, right? You're looking, or corporate money in this case. You're just a fishing expedition. What are you going to get out of it? And of course, you're going to talk in a moment about exactly what you found.
So I think the question really with fishing expeditions is whether or not you're using a hook and one type of bait, or instead hedging your bets so that you can look at an outbreak. Whenever you have an outbreak, the probability is much higher that you're going to find something because there's going to be many. There's going to be theoretically one agent, affecting all of these animals. So it was a very, very good bet. It wasn't like playing the lottery. This really is much more likely to pay off.
Right, and you also, I guess, this wasn't so much a baited hook as a trawl net, right? You did a very open-ended type of approach.
Yeah.
Besides, as we discussed before, this is not a fishing expedition anymore. It's discovery-driven science.
That's right.
Exactly.
Besides, some of us know how to fish.
So, Amit, what was the first thing you did? You had these respiratory samples.
Yes, so
do with them?
So we what we did, we enriched these samples for viral nucleic acid, and how we do it actually, we treat sample with nucleases, we centrifuge the sample and pass through filters that will let only virus go through them. And then we actually try to amplify everything that is there. So it is unbiased amplification using random primers. And in that way, you expect to find whatever is there without being, and then you send these samples for some kind of high throughput sequencing. In this case, we use 4x4 technique because I think Ian Lev is the one who started using 4x4 to do these things. So we were very lucky to have resources, and we started. Then we have a bioinformatic group that helped us analyzing all this data. And we found other viruses too, but this was the one we started focusing on and trying to chase down the complete genome.
So I'm wondering, what size filter did you use? A 0.2 micron?
No, actually, we go for 0.45 micron because I believe if there is like antibody-bound virus or sometimes there are clumps, then we may lose some virus during filtration if we use 0.2 micron.
Do you think there's some bigger viruses that you missed, though?
Yes, means generally we are not expecting something like pox or herpes, but they are in that range. So, and when we are talking about these filters, they are not exactly what they say. So if they are saying they are 0.2 micron, it's a range, it's a mean actually diameter. So you have some small pore size. So there are something, I talked with this company, they are somewhere between 50 to 300. So we chose the upper limit of actually 450 micron filter, so that we will not lose any virus particle during filtration.
You just want to make sure you get rid of the bacteria, really, right?
Yes, and bacteria are generally more than 1 micron. They are generally 2 micron, but if you go to the smallest, they are like, the mycoplasma can be a rare exception, but we don't want to lose virus, and if there are some bacteria that we can, amplify, that's fine for us.
The key to the success with any of these approaches is sample prep. So you have a lot of extraneous material. You need to remove that because the sequencer is going to give you a view of everything. So you're going to dilute out the signal, or you simply won't see it at all. Thus, the nucleases are critical. The filtration is critical. Centrifugation is critical. And then, as Amit say, you get a snapshot of everything that's present within a sample. Now, the earliest successes, you probably remember, with these sorts of unbiased sequencing methods when sample prep was a little less well advanced, was with material that came out of supernates of cell culture or
Right.
acellular fluids like spinal fluid or serum. So when you're working with something which is coming out of the respiratory tract of a dog, there's a lot of things in there that can, that can confound you. So this is very, very helpful in cleaning it up.
Yeah, and I should add that even after doing all this, we still see a bulk of amplification from bacteria and other. So we can't get rid of.
So what percent of the sequence is viral?
It actually really depends on the sample, what kind of sample you have. Sometime about 60 to 70% of total sequences are derived from viruses. But generally, the trend is like 20 to 25% virus in a good sample, and rest all is bacterial sequences. Some may be coming from the reagents itself.
So even healthy dogs have viral sequences?
Actually, we didn't look. That will be true fishing. But we didn't look in actually normal dogs for their flora. But I expect, like in normal humans, they will have something.
Well, but this background level of noise of bacterial and other sequences that you see in there is actually good in a sense, because it means that you probably didn't exclude too much, right?
It's good, but I think it's more bad for us because it's
an annoyance, certainly.
Yeah, it's a noise, and it's actually it's reducing the total amount of useful sequences that we can get from the procedure.
So do you actually put in known quantities of known entities so that you can test the system to make sure it's working?
Well, in the early days, when we were developing this technology, we piloted, performance with various modifications. At this point, these are such expensive assays to perform that you focus them and you look for candidates. And once you have a candidate, then you go back and you test for specificity.
Okay.
So, but we do have insights into sensitivity with and without various modifications. The other thing that's important is there are many sequencing platforms now. Some of them have, can give you many, many more reads. The reason we still like this one is that you get long reads. It's much easier to find something that's going to be reminiscent of an agent, particularly if it's really an unknown agent.
Oh, okay.
And by long, you mean how long? 100 nucleotides or so?
Much more than that.
So by 454, on average, we get like 300 to 350.
Oh, okay.
Yeah.
And that helps you overlap them as well. Right?
Yes, it helps them overlap and make longer contacts, something we call,
Because with these like PacBio, my understanding is you get 30 nucleotide runs and then...
No, PacBio is giving you quite long runs now, but the error rate is a problem still. I'm sure that'll come under control.
Some of them give you 30?
Some of them are much shorter platforms.
Yeah, the Illumina sequencing is short sequencing, right?
And then you have a real problem overlapping.
Well, there are a number of mathematical games you can play, but it's still not where it needs to be. Yeah. So it really, these are all tools, and it's a question of the job.
But the proof is then to go back and isolate that entity that you're interested in.
Well, maybe not isolate the entity, because that we've not achieved yet.
Oh, okay.
But to get additional sequence, yes.
All right.
So, Amit, you initially saw traces of something that looked like a hep C? Is that how it ran?
Yes. Actually, it was we got initially 3 or 4 sequences that were looking like by sequence similarity to hepatitis C virus, but they were very different. So unless we had like about 2,000 or 3,000 nucleotide long sequence, I was not very sure that I should say something to someone that we found something.
Right.
Because it was really...
Look what I caught.
And
so you mean you didn't tell Ian right away?
Yes. No, but
you might have just hauled up a boot.
No, he was
exactly, or an old tire.
Yeah, but he was the first one to know after I was sure that it is something similar to hepatitis C, but it was also giving showing a strong similarity to other hepatitis C-like virus like GBVC and others. But once we reached the envelope of this virus, this new canine hepacivirus, we were very sure, because hepatitis C viruses are very diverse in envelope. And particularly it is very different from GBVC virus. But canine hepacivirus, it was consistently more similar to hepatitis C.
Now, if you start out and you get a hit, but the sequence is really pretty different, it strikes me that it's really a challenge to develop primers or whatever to extend the sequence because you don't know what you're looking for. How do you approach that?
Yeah, it is very challenging. Actually, viruses that are very diverse, they are very interesting for hunting. But I think it is very challenging. And in that case, what we do, we actually make an alignment with all the sequences or all the viruses that are of that family, and then try to design consensus primer and extend the sequence. Yeah.
Okay.
There's another thing that can be very helpful. If you think you know what sort of a virus you've discovered, you assemble a scaffold, and then you go through all the remaining sequence that you have from the run, and you try to see if you can patch it together in some way,
right.
And then you span the gaps between them, and that can also be quite helpful.
Right.
Did you guys ever think of collaborating with the group up at the Baker Institute? Because they actually have the canine distemper virus vaccine developed. There's a lot of other things that go on up there. I knew the guy who used to direct it. And I don't know if they were ever involved in virus discovery or not, but they'd be very interested to hear what you guys have come up with.
We are working with people in Cornell now, but on follow-up study of this virus, because now we are interested in determining how common is this virus. And I'm just on a general point, when you find a virus in an animal, it is not just by nucleic acid techniques, it is not very convincing to say that this virus is actually a canine virus.
Right.
So what we are trying to do now is to gather serological evidence, because this may be an unexpected case where these animals somehow got this virus from
the drinking water.
No,
that was a joke.
Yes, but
right, because all your samples came from the same place, right?
Right.
A same shelter. They are different animals. Yes.
Yes, but we've found them in other shelters as well. So.
Okay.
Actually, I have one more brief.
Well, let me just emphasize something that Amit has said, because that's critical. Finding a sequence doesn't prove its presence, and it certainly doesn't prove relationship to disease. And serology can be very helpful. I mean, that goes all the way back to Thomas Rivers, who talked about using adaptive immunity as a key to understanding relationship to disease. So, and it's important also to emphasize that the genetic methods have really far outstripped what we can do in serology. And this is something that we're trying to tackle now, develop new platforms that can be used for discovery that are based on immunological responses.
But in the end, you have to get a virus from any host to show that it's causing disease, right?
Yeah, that's why they call virus first and disease next. So at least we can get the sequence information, and then we can design all these. Means we can express these proteins using that information and then try for.
I don't know that that's true, Vince. I think it's largely true, but not invariably true. There are instances where I think you could, and maybe these are still hypothetical, where we may not be able to grow a virus, but we can demonstrate that we can control it in an animal model using something that's specific for that virus, an antiviral, for example. So if you have very strong epidemiological data, very strong data that comes from vaccines or antivirals, that may in fact replace the need to purify the virus in culture. And maybe you can get by with just using, something like a sputum that has not really been really refined.
But you do try and isolate a virus.
Oh, God,
right?
do we try.
And so in this case, you have tried to isolate virus by putting these nasal samples onto cells, right?
Yes, we did that. We actually, unfortunately, there are not too many cell lines from dogs available in ATCC. And we used actually one cell line that was there, and then we also used one primary cell line. But we were not able to grow this virus.
Have you tried infecting another dog with these samples?
No, not yet. And the problem is that the samples, they were not enough, and we don't. Sure. So if we
I was going to ask what does a respiratory sample consist of? How is it collected, and how much do you get?
So I can tell you about these samples that I've worked on. So what we did, what we asked them to do is actually take a nasal swab and put it in 3 ml of minimum essential media. Or you can also use viral transport media or even HBSS with antibiotics to stop.
So it's just a swab. You're not talking about much material.
No. It was not something like, bronchoalveolar lavage or something.
Right.
Yeah, it was just...
Okay.
If you want, Rich, you can go and lavage those dogs.
No, thanks.
Yeah.
So you did try to look in these infected dogs or in these dogs who were sick for viral sequences, right?
Yes, we did. And so after seeing this sequence, we extended the genome and then we designed a specific PCR assay to look for this virus in all the related animals that were from there, that were grouped in this shelter. And we found in few animals that were related, they had similar kind of symptoms. We also looked in different tissue samples from unrelated animals to see, to determine the tropism of this virus. And what we saw is that we found few liver samples that were very weakly positive. So to confirm these results, we did in situ hybridization.
Mm-hmm.
Oh, yeah.
And that what is shown in this.
This figure 1 in your paper, right?
Yes.
So this is sections of liver from some of these sick dogs? Is that correct?
These are 10 micron sections of liver from some dogs that were PCR positives.
So I remember that Ian and I met shortly after he came here because I wrote a small book about the West Nile virus, and I featured the work that you did out in California by sequencing the virus out of the tissue itself. Have you tried to get sequence out of this tissue?
We didn't actually, because this tissue was we did quantitative PCR assay, and there was very low titer of virus present in these tissues. So we didn't try that.
The sequence came from the respiratory sample.
So our strategy, whenever we're trying to do this kind of work, is after we get the first piece, the tail, right, then we establish a quantitative assay, determine which is the best source for template to do the sequencing, and focus on doing the sequencing from that particular source, which in this instance was the respiratory samples. Now, if the liver had been higher, then we would have gone back into liver. The in situ hybridization is a shortcut that we've taken because it means that we don't have to make antibodies, which can take a very long time. It takes months. You've got to make protein, and then of course you have to immunize animals and get antibodies. So this really cuts the time down. So you can go directly from sequence to in situ hybridization in 3 or 4 days. It's a very rapid process.
So how much virus has to be in a tissue before you can get a decent signal from it to make a sequence from it?
I mean, in this case, it was like 100 virus equivalent in the about 10 to the power 5 cells. So that what we observed in this case, but it depends on really the starting material and the extraction method you're using.
So for the West Nile, just by comparison, what was the viral load in the tissue that you used to get your sequence?
Oh, it was
massive.
We never
because they died from it, actually.
Well, they died from it, and postmortem specimens are typically lower in viral titer than obviously because there's nothing to support the growth of the virus.
Sure.
The best example I can give you would not really be West Nile virus, but some work that we recently did with an astrovirus that came out of an immunosuppressed child. And in that instance, the premortem sample was 10 to the 7th copies, and the postmortem was 10 to the 4th. And that's a fairly reasonable estimate of what happens when people have a slow dwindle. You see loss of several orders of magnitude.
So what's the sensitivity for your in situ?
Means we focused on, so it is like focal infection. Mm-hmm. So we were looking for many spots, and we were trying to find where we are getting signal from.
Right.
So in terms of sensitivity, we didn't really know about the in situ hybridization assay, how much sensitive it was. All you can do is look for sections and see the signal. But these are all PCR confirmed, and we got sequence information from these cases because we sequenced that PCR product that we got to confirm our results. And
so where else in the dogs did you look for?
We looked in lungs and spleens. And we didn't see, but maybe it is further in lower titer, means more, even lesser than it was in liver.
You mean
samples.
You looked in sections of lungs and spleens, right?
No, we looked into actually extracted material from these things. So this in situ hybridization method was not used for screening.
Sure.
It was just for confirming our PCR assays that we did.
But in the respiratory samples, you went back and measured copy numbers there.
Yes, we did
and
that.
You could detect RNA, viral RNA there, right?
Yes, and we used a nested PCR assay. It was very sensitive, up to 10 to 100 copies per input.
And did the liver sample itself give any cytopathology that would indicate that they were suffering from hepatitis?
No, there was no sign of that.
None.
Yeah, they were normal.
The in situ hybridization really kind of tells you that there's actually an infection going on.
Yeah, right.
That's what we wanted to determine, because we thought just by saying by PCR that we found those sequences, we will really don't know if it is just the fluid circulating in liver or it is the cells that are infected.
Right, but this way you can see viral RNA in the cells.
Yes,
yeah, right.
yes.
So if it's a virus of dogs, it's not a very permissive system.
Yes, it's not,
So,
Well, not in liver.
Yeah, that's
Not
what I
in
meant to
liver,
say.
yes.
Not in liver.
I mean, these dogs had respiratory illness, not hepatitis, as far as you knew, right?
Correct.
So that might have lowered their resistance and allowed this to happen, do you think?
We don't know yet. I should not say, we don't know, and
A lot of interesting questions still.
no, and it's just the beginning. I think we still don't know if it is just a latent infection, how this, what is the nature of this virus infection.
But it's important, though, because if dogs are the carrier, could be a carrier for human hepatitis C virus, that would
that is very unlikely.
complicate a big issue here.
Yes, yeah.
Do
Ian's
dogs
waving
get
his hands.
hepatitis?
He's waving his hands. Please don't go
It's
there.
very distant from human hep C.
I mean, how different is it? I guess I should have asked that first.
Well, we can talk about the history next, but before we do that,
Sorry.
and I just want to emphasize that we don't think there's any possibility at all that these dogs have transmitted hepatitis C to humans.
Okay.
And this was a concern we had when we were discussing a release on this paper.
Right.
And Pfizer emphasized this as well, because if you think about it, people might use such arguments to say that we should kill all the dogs in all the shelters.
Exactly.
Right. So this is a very important point to make to your audience.
But the other way is to look at it, maybe they caught it from humans.
Now that seems also unlikely. But just to finish the point about the steps in linking diseases and agents. So the first step, of course, is to identify a candidate, get as much information about it as you can. Then you try to get some quantitative data, which can then be helpful in thinking about where you might want to do in situ hybridization or immunohistochemistry to look for localization by organ. And as a good rule of thumb, if you don't find it by PCR, it's unlikely you're going to see it by in situ hybridization in our experience. Though it's possible that there may be rare cells that are infected, and there's certainly some herpes viruses that will do that, particularly in blood. Then, of course, you have to go through the rest of it, right, which is the serology and trying to develop models and interventional strategies. So we're on the road, but we're certainly not to the end of the journey.
So you're interested in staying on this road, though,
we're very interested in staying on this road.
So what cell culture technologies would you employ to look at extracts from these infected dogs?
So what we are doing, we are trying to develop some primary cell lines. And for that, actually, so we are collaborating with people in Rockefeller and Cornell and other people who are interested in this viruses. And we are going to approach it in two ways. First, we want to find out by screening more animals that if we can get a sample with high titer virus, so that we can use that virus to infect these cell lines. The other way we are approaching is actually we are making pseudotypes using hepatitis C backbone. So we are trying to express the envelope protein of this virus, canine hepacivirus, on hepatitis C backbone. And also using lentivirus vector for that purpose. So the purpose there will be to determine the cell tropage so that we will be more focused in our attempts to grow this virus.
So you take the envelope of this canine virus and put it onto other particles.
Yes,
and infect with those.
Yes, and this work is actually going on in Rice Laboratory.
Mm-hmm.
Mm-hmm. I meant to ask about the dog types also. I mean, there are about 400 different breeds of dogs. Which breeds were in this shelter that might have certain some differences if you'd done them individually rather than all together?
Means the most I know, the most information I have about these animals where we found this virus is that they were not from same breed, so they were from different breeds. So it is not restricted to one breed, and we really don't know because they were not,
right,
samples.
The other approach you can take is to...
They're mutts.
They're mutts.
They're mutts,
Right.
just like us.
Just
This is
like
the whole
us.
room of
Yeah,
mutts
we're
right here.
You can make an infectious DNA copy of the genome and try and get
Right.
virus from that.
Yeah,
right?
sure.
It's very hard. Like for hepatitis C, it took many years to do that, but we will, because of hepatitis C data, HCV replication system. I think we will be able to do this for this virus, but in parts. So we will try first to generate some kind of recombinants between hepatitis C and canine hepacivirus, and ultimately when we will have this virus growing in cell and we will be able to find out the cell tropism, then it will be worth trying making an infectious cDNA clone and these things.
We're also not clear on one of the termini.
Okay.
Yes.
You don't have one of the ends.
So we are missing the 3' end. Actually, we got something, but it is so drastically different from all we know about hepatitis C that we are not very sure. And that's why also it is key to get a sample that is high titer and positive for canine hepacivirus.
That 3' end was an issue with hepatitis C too, wasn't it? Getting that right?
It was an issue for many years, and Rice Laboratory is the one they were successful in getting it. It is very unusual in this viruses because generally all you see in RNA viruses is either poly A tail or no tail, but in these viruses you have a poly T or C stretch, and then you have a unusual 3 UTR. That is.
So how are you going to get the sequence of the 3' end? You're going back into the dog material?
No, we got something. It is not that we don't have anything. Okay. We tried and we used RACE, rapid amplification of C, and then we also tried an adopter ligation approach. So we tried both the approaches that were quite successful in getting the hepatitis C virus 3' end. And we got something for this virus, but I'm not very sure about the sequence because it is very different from hepatitis C virus. Unlike the 5 UTR, which is very similar between these two viruses, and which is So your problem is attaching this extra sequence onto the 3' end of the clone. That's where you're having difficulty. Yes.
Okay.
Yes.
Well, you'll solve it. It's a technical problem. You're skilled. You're highly skilled.
All we need is a high titer sample again.
Have faith.
Inshallah.
Yeah. Thank you. Yeah.
So now you're looking at a bunch of additional samples to try to find more sequences, right?
Yes, and we are trying to actually get samples from all over the world, not only from US. Sure. We have now new links in Germany, Senegal, and other places where we are going to get dog samples, and we will look for this virus.
Go to Bhutan. But I'm not kidding. I've been to Bhutan, and there are more dogs in Bhutan than there are people.
Yes.
There
Are
are
you gonna check your
unbelievable
dog,
numbers
Ian?
of Bhutan.
And people eat their dogs too.
Not in Bhutan.
Not in Bhutan?
No, they're vegetarians.
So you're going to get respiratory samples, or can you look in? You would like to look in other tissues to see the prevalence, right?
Yes, we are looking for now respiratory samples, but ultimately we want to see if we can get a serum samples that is positive or a tissue sample that is positive, because actually that will help in also getting the 3' UTR, because if you have negative strand RNA, then it is easy to get, and that only you can get in the cells or in tissue.
You should probably also find a little more diversity in the sequences as well, because they seem fairly uniform in this paper.
Yes, and I mentioned in the paper that it may be due to the animals that we had samples from. They were related because of this outbreak that was going on, and also because of the primers we were using. But now, expecting like hepatitis C or like any other RNA virus, we are trying to develop, and actually we developed a PCR assay that will pick up anything that is similar or that is in between hepatitis C and CHV or canine hepacivirus. So we will cover all that diversity while we will screen all these samples from different sources.
Right.
So,
Do dogs get hepatitis?
Yes, no, I mean, I don't know if because of this virus or any other virus, but hepatocellular carcinoma is very common in dogs.
Mm-hmm.
Hepatitis is actually common in dogs as well. There have been a number of people with whom we're trying to work on that, and but this, in fact, some of the people said, "My God, why didn't you tell us that you'd already found this before we sent these samples?" And in fact, we were not looking for hepatitis, and we don't think that this particular virus is a major cause of hepatitis in dogs.
Okay.
So that's still an open question.
So you don't think it's a major cause of hepatitis in dogs because you found it more abundant in the respiratory tract than in the liver?
And because we've gone back and looked for this particular virus in other samples, or at least other people have, with whom we've worked, and they haven't reported to us that they're finding it.
Yeah, but it is too early to say. And the other thing is that in dogs, generally, hepatitis is caused by adenoviruses. And so in most cases, it is known. But we still don't know how the hepatocellular carcinoma develops in dogs.
No, we don't.
And that is interesting. But we don't know if this virus is even hepatotropic. So it is very early to say anything.
So this gets back to nomenclature. We've called it a canine hepacivirus, right? But if it's not a hepatotropic virus, is that the right name for it? Another question.
Right. Well, so yeah, that gets into the nomenclature of that whole clade, okay? Because hepaci is, I mean, that's a bioinformatic grouping of something where the major virus is a hepatitis virus, but it may ultimately include viruses that are closely related but don't cause hepatitis, right?
That's a problem for the ICTV.
The flaviviruses don't all cause yellow fever either.
Yes, that would actually...
Just to throw my limited viral knowledge out on the table here.
No, yeah.
Someday we're not going to give them descriptive names anymore, just numbers.
I think we're probably stuck with the descriptive names.
Well, rhinos are enteroviruses now. Right?
They are in that genus. But
they're put in the genus, but they don't cause enteric infections. And people don't seem to worry. I worry about it.
Well, if you look at the respiratory tract as a contiguous epithelial...
Dickson, but the respiratory tract is not contiguous with the alimentary tract down here. At least mine is not.
No, but it starts out the same.
Yeah, I know, but that's not a good argument.
I don't have a good argument.
It's a mucosal surface. But in people...
Yeah, mucosal surface, that's what I meant.
Yeah, that's a good question. So you had mentioned something about history we wanted to get to. Before you leave, you have to leave in a few minutes.
To take care of my dog.
Yeah.
Right.
Okay, that's a good reason.
Who may be suffering from
How
hepatitis.
old is this virus? Does it predate... it predates the discovery of hep C in people, obviously, right?
Yeah, so we did evolutionary analysis to find out that if we can determine when these two viruses diverged from each other. And we used an approach that people generally use to date the evolution events. And we use the evolution rate that is known for hepatitis, because we only know one canine hepacivirus sequence, so we can't use that. So we use the rate, the substitution rate or mutation rate of hepatitis C viruses. And we used, now in hepatitis C, there are huge diversity. So there are different genotypes and they evolve at different rates. People determine that. So we use the same rates for two of the genotypes, 1 and 6. And we try to date the emergence, if we can say so, from one virus to other. And we don't know which one emerged from which, but we try to date that, and we estimate that it will be about 500 to 1,000 years. But these methods are just theoretical, and they are better if we are talking about viruses within a species. Now, if these are two species, we really don't know that. So we tried the best that was there available to date it.
And one of the things you pointed out in your paper is something we've run into before, where dating based on the substitution rate in known viruses gives you a very different answer than if you date based on sequences that have been frozen by integration into the genome. If I understand this correctly, you can get really different numbers. So it's a tough business.
Yes, that's why I said there are several factors, like you said, endogenization. And there are recent reports that suggest that not only retroviruses, but other viruses also became endogenous. In one of the papers, they mentioned that there are some flavivirus-like sequences. So if according to these dating techniques that we use, just based on substitution rate, we really can't say something. But
yeah, you're very careful in the paper to point out all the caveats.
Yes,
right.
I mentioned that, and I also cited some references that suggest that these approaches don't work, particularly when you're talking about very diverse group of virus. Now, these techniques are very good. On the other hand, when you're looking for expansion of an infection like hepatitis C, how it expanded in human population, and there was very nice work reported by Pybus and others and from UK. And we use the same rates, and that all was there to use. So, yeah.
So does this give clues about the origin of human hep C?
We don't know yet. I think more important will be to determine the real distribution or one can say ecology of these viruses in different animals. So once we will have that picture more clear in terms of diversity, means if some related viruses like canine hepacivirus or even human hepatitis C virus, they are infecting other animals of canid or felid or other carnivore animals, or even non-human primates, then we will be able to conclude or reach a conclusion about.
Right. Are you going to look at that in other animals? Are you looking?
Yes, we are trying to get some samples from foxes and wolves, but this all means I think it will take a year or so to completely.
They're a little harder to deal with than domestic dogs.
Yes,
Cats.
and we are also trying to get wild dogs to see if it is an old infection in dogs, because now people are raising question that what about reverse zoonosis, like some of the listener of TWiV or asked on the blog. So I think we are looking, but for now we focused on more the evolutionary implications and the comparative genomics of this virus rather than reaching a conclusion.
It's interesting, though, to speculate in that direction, because I think Dickson suggested, how do we know it didn't go from humans into dogs?
Right.
Certainly possible. It's also possible that there was another animal that gave rise to this infection in dogs and humans, and we can't really distinguish between them. Now, you're in Texas, right?
Yeah.
Now, Hines Cave is not too far from Austin, I think, and that's where we found evidence, not we, but the others found evidence of dogs in a cave that were apparently consumed. They were in human excreta.
Oh.
So man's best friend was not treated so well about 8,000 years ago. And it is interesting to speculate that it might have been some sort of trafficking, interspecies transmission at some point.
But we know people have not done virome analyses in wild animals to any great extent, right?
Well, when you say to no great extent, that I have to agree with that for sure. But there have been a lot of studies now, or some of them published, some still coming out, where people are trying to do precisely that. So we've certainly seen a lot of bat studies,
Bats,
rodent studies. We're doing similar things now with companion animals. We're doing this now with large primates, different types of apes, monkeys, and these will be illuminating.
But no one has so far found any sequence that's like your canine hepacivirus.
Certainly not. Yeah. There is no report on such sequence.
So before this, the closest relative that we found was in a bat. In fact, this was work done by John Epstein, Eco Health Alliance, where he found this GBV-like virus, and that was about a year and a half ago. So it was ironic, right, that the next one that comes up is closer yet. So next week, maybe we have something even better.
That's what I mentioned in discussion, that these two group of viruses, hepatitis C virus and GBV, they're very close.
Right.
But they are very different in terms of their evolution, because if you look at human GBV-C virus, you see many related viruses in all these animals like non-human primates that are very similar to human GBV-C. So that's why we thought that this virus may be more common type of primate originated virus. But on the other hand, for hepatitis C virus, there is no homologs in non-human primates. And this is the first one. So that's what makes it more interesting. And like I said, we were not looking for this virus, so we also found some other viruses in these samples. that are most closely related to human. And one of them is actually a picornavirus, a human virus called hepacivirus. We don't know about the origin of hepacivirus, similar to hepatitis C, because it is very different from any other Kapoor virus. But we have a canine virus now, and it is in review now. It will come out soon, where the story is same. It is most genetically related to human hepacivirus, and this is a canine virus. So
they're from the same dogs as this study? Yeah.
They are different animals, and they are gastroenteritis, means they were suffering from gastroenteritis. So means I think this group of animals, companion animals, like we said, they are very important. They were never studied in that detail. Means people were interested in finding their pathogens, not the normal viruses that are there, and they may be related to human virus. Because we must be exchanging our flora with canines for a long time. So
people who have dogs really get close to them.
Right. Yeah, they
My
do,
son is always sleeping with his bloody dog.
Yeah.
I tell him, that dog has a lot of microbes, and you know what he says? So do we, Dad.
Yeah. But
good for him.
You brought him up right.
There you go.
But in that instance also, when we did evolutionary analysis in this hepacivirus related virus we found in dogs, what we see is that if they are somehow related or emerged from each other, it happened long time ago.
Right.
So they are not that closely related that we term them recent zoonosis or reverse zoonosis. It happened very long. Or it may be a situation like a SARS virus where you found it in cat and you thought it's coming from cat in humans, but then we realize it is coming in both from bats. And so it may be we will find in future.
Sounds like you have plenty to do with this virus.
Yeah.
But you're still doing other things as well, right?
Yes. Yeah. For now,
That's
yes.
the way it goes. So before you leave, Ian, and in this paper you start off by saying viral zoonoses account for up to 70% of human emerging infectious diseases. But when you look at all the human viral infections, aren't they all zoonoses, really? Or do you think some of them emerged in people without coming from an animal at some point?
Good question.
It's a very deep philosophical question. Unless you believe in the creation theory,
right, that's
where
right.
humans just appeared, right?
Yeah.
By definition, other things preceded.
There you go.
Right.
That's right.
So those other things that preceded them had infections. And they passed them on.
Presumably so. And I think you alluded to Ann Skalka's paper recently, where she was talking about flaviviruses and filoviruses and bornaviruses and all sorts of other agents. And we have some other ones where we talked about genomic integration as well. Many of those events have been useful in dating, the evolution of species.
Sure.
Because we can actually, it makes it possible for us to get back to anthropology, which is where I started.
Yeah. You see that? You were prescient.
So creationists believe in the born-again virus.
Yeah.
Dickson.
Alan, that was just for you.
That was supposed to be my job, yeah.
It was a poor attempt.
All right, Ian is leaving in 2 minutes. Are there any final questions for him?
Wait. When he walked in the room before we started this broadcast, he said, the man sitting next to me to my left said that his next project is to sample the bottom of the Marianas Trench for the virome of
microbiome,
the very bottom of the earth.
He's going to
go. We've got some
as
really
deep as you can go.
There's so many interesting projects to do. We're doing some work now with the Yanomami Indians, an isolated group that was found in a flight over the Amazon about a year and a half ago.
Oh, they've been known before that.
No, but not
this
particular,
group of Yanomamis?
not this particular isolated group.
Stand corrected.
And there's some very interesting findings that are coming out of Marty Blazer's group and Gloria Dominguez. I mean, it's really fantastic stuff. Very, very restricted E. coli speciation. And we're looking now at particularly at herpes viruses. Those might be interesting. The one problem with the notion that this is an isolated group is that some of the individuals in this particular tribe happen to have blue eyes. And so presumably there's been some mixing somewhere.
There
And
you go.
you also have to get to them really quickly because I don't imagine they're going to be isolated for long.
No.
They don't
True.
like to be
True.
mixed with different people, though. They're sort of fighting that deal, I think.
Right, but if researchers are going in, then by definition they're not isolated anymore.
No, I understand that. They have an interesting farming system that I talked about a little bit.
It's not vertical, though, Dickson.
No, it's not. It's circular. It is circular. They clear an area of woods, they take all the byproduct from their chopping down, and they burn it all in one little place, and then they farm it until the nutrients run out, and then they move to another circle. And by the time they return to the first circle, it's about 30 years later, the forest is totally regrown.
On that note...
You'll come back and tell us more about these broad projects, okay?
I just want to tell all your listeners that there's a spectacular view of the George Washington Bridge from Vince's office.
Don't you have that view from your office?
That's fantastic.
Well, but I'm not under scrutiny here, Vince. You are.
Okay, thank you, Ian.
Adios.
Okay.
Take care.
See ya.
I'm sorry, did I mention that Kobu virus and Aichi virus, right? So it doesn't matter because we are talking about hepacivirus, and I should have not mentioned picorna or...
Right, say what you like.
I was just making a case that these animals may have a lot more than...
You can say whatever you'd like.
You've got the floor.
Yep. We talk about all sorts of viruses on this show.
And you can
Exactly
see Dickson.
right.
Dickson is talking about farming.
Yes.
Of all things. You can do whatever you want. It's no problem.
As a
One
matter
track
of fact,
minded.
that was one of my first questions I wrote in here because you set out looking for picorna viruses. I was wondering if you found any.
Yes.
Apparently you did.
Yes, and it's in review actually. I hope they will accept it. It's exactly same as this paper.
That's great. I think it sounds like we're going to have you back again.
So the Mariani Trench really would be a fishing expedition.
Yes,
Indeed.
Would you like to go to the bottom of that trench?
I would.
Yeah,
We've actually gone there once.
Oh, yeah?
Yeah. I think
What do you mean we?
The human...
Oh, I thought you meant
Humans.
you and someone else.
Humans have gone once.
How
Okay.
deep is that?
It's 36,000 feet.
Wow.
It's as deep as it's deeper than Mount Everest is tall.
Is that the deepest part of the ocean?
It is the deepest part. It's off the coast of
So that's like 5
Philippines.
miles.
It's 5
Yeah.
miles.
It's as high as I
I
fly.
would go, but I'm not sure I could handle the pressure.
Oh, Alan, you can.
Okay, let's see what we should do here. I think we've talked enough about this, right, everyone? Amit, did we cover everything?
I mean, there's a lot more to talk about, but...
I think we
you
covered
want to
it.
change
Why
the
we'll have him
subject...
back on the show.
Exactly.
We'll have him back when he's
Exactly.
got more on this. And this
Yeah.
is just great. There's so much to do.
So, what I did want to say, though, I want to inject something here because what you're doing and what Vince asked Ian about, are you going to isolate the virus and then try to reproduce the infection? Okay, so there's an analogy here that began way back in the history of science. And somewhere in I guess during Madame Curie's reign in France, I just finished going to the World Science Festival and I saw the play that Alan Alda has written about Madame Curie, and it was written up in Science magazine, by the way. She received 2 Nobel Prizes. Okay, the first one she received for discovering polonium, and she did that with her husband. And her husband was given the major credit for this, and she was looked at as his technician. Which insulted her because she had to sit in the audience while he received the Nobel Prize, although they both got it.
And he acknowledged that?
Of course he did, but he... Yes, he did.
Yeah, he had to go home afterwards.
Yeah, the point is that they didn't purify it, okay? And then what they also discovered was that there was another element besides uranium and polonium in this large sample that Becquerel had left over after he discovered uranium called radium. Okay, which is another element.
Yeah.
Okay, it's a decay product, all right? But it was in such dilute amount that no one wanted to work on this except Madame Curie. So then her husband gets killed in essentially an automobile accident, but it was actually a horse and cart that ran over his head and smashed him one day, and he was dead. She continued her work, and she eventually purified radium from this enormous pile of tailings that were derived from this uranium mine. And she isolated it in pure form. But until then, no one believed that the radium existed. She had to actually prove that it it was purified to the point of being a pure chemical, and she wore it in a little locket around her neck. And of course, that's what induced her thyroid cancer.
Yes, she did pioneering work on the carcinogenicity of radiation.
That's right. Now, the interesting part is when Alan Alda went to look up the papers that she wrote, she was also involved in a tryst and all kinds of other interesting things. He had to get special permission to look at these letters that were all radioactive still.
Yeah.
The letters themselves had lots of radium all over them. The point is that no one would believe that radium existed until it was actually pure.
Yeah.
All right, so to make that analogy here, we would love to see a pure virus. You would too. I know you would. The particles would be purified and looked at in an electron microscope. You could reproduce the infection. Even though it's low permissive infections in dogs, there may be another cell line which would allow this thing to expand out and really take off, right?
Yeah.
So,
No, we are looking for that, and if suppose we find some high titer sample, we are going to do this, and
Sure.
and just as a foundation work, what we are trying to do, use these pseudotype viruses, means these lentivirus vectors that are expressing the envelope, to focus our efforts on that cell line that will support ultimately. And so we will determine the cell tropism first, and then try to grow this virus.
So how much work, like I'm not a virologist, and a lot of the listeners are not either, but they're interested in the subject. What does that kind of work entail, and how long down the road do you think you have to go before you give up, or you say, aha, I found it?
Means you mean the follow-up work or the work?
No, the follow-up. The follow-up to get the pure virus.
It depends on how interesting it becomes.
Okay.
Means if suppose we will find in 1 year there are a lot many viruses like these, then it will be important for evolutionary perspective. But I don't think people will just focus on this virus and they will try to pick the best one out of these. But if this will be the only one that remains, the hepatitis C homologue, then like we started it with Rockefeller people, they are trying all different types of, because I mentioned in this paper an important point in hepatitis C, although they are very different genetically, all these genotypes, but they have this MIR 122 binding site in 5-UTR. This is absolutely conserved. And in this virus, canine virus, it is not there. So now Charlie Rice group, they are trying to make hepatitis C, and this MIR 122, it helps hepatitis C to replicate in liver to high titers. So what this group is doing in Rockefeller, that they are mutating hepatitis C site and making it like canine virus to see if it can replicate in lung or other, so to change the tissue tropism. So what is more important about this work or this kind of work where you are finding all these viruses, I think, is that no human or no technique can predict what will be a difference between virus from one host and other. So that you can only know by discovery, means by finding new viruses, and then you have this information that you can use it for like determining replication, pathogenesis, and these things. So
so it's a lot of work ahead of you.
But I cannot do all, and
absolutely.
I'm sorry.
It's good to have work, right?
Oh, no question about it. No, I think one thing leads to another. That's the whole deal.
At some point, Amit wants to go off on his own, and he'll have plenty to take with him. Obviously.
Oh yeah.
Although it's a very attractive situation here because you have so much support. There's bioinformatics, there's sequencing, right? And
that's right.
To do that all on your own is very difficult, right?
Not really nowadays.
No, that's not,
because now people are developing all these softwares that you can do all this analysis. Means like when I was in Eric's lab, means we were like a small group there, and I know you went there last year, but means I started like 5 years, we were like 3-4 people, and we did this.
You did it, that's true. He does a lot of outsourcing,
Yes.
Let's do some email, but before we do that, we should just mention that there was a very interesting paper this week from the PLOS group at PLOSS at Rockefeller. It's in Nature. Who put this up here? You, Alan?
I put that up there, yeah.
A genetically humanized mouse model for hepatitis C virus infection. So the first author is Marcus Dorner, and the last author is Alexander Ploss.
And Charles Rice is also on this paper.
They're trying to develop this mutantized version of hepatitis C a long time.
So I think we should do a hold a separate TWiV on this to talk about it. 'Cause
I
it's
agree.
very interesting.
Yeah, this is a very cool piece of work.
In fact, we could probably get Matt Evans to come up and do that with us.
Good idea.
So I just want to mention it's an interesting mouse model for infection with Hep C, and there's some interesting aspects of it, but I don't want to go into it right now.
Nope.
Just to point it out. All right, let's do a couple of emails. The first one I put up here is actually from my blog. I'd written a blog post about your virus, Amit.
Yeah.
And it's from Alex, who said, although I'm sure there's a good reason to disqualify this possibility, why can't Hep C be a human-specific pathogen that transferred over to dogs? You don't specifically mention this possibility, so I'm genuinely curious. So, as we have said, it could be, right?
It could be. It could be. We cannot rule out anything. Yeah.
Right.
All right, the next one is from Chris. Dear professors, I love the TWiV, TWIP, and TWIM podcasts because they are entertaining and I learn so much, even though I've only taken one biology course in my life. I enjoy listening to all of them as I garden.
Really?
It is springtime, and he's in Seattle, and the tulips in my yard are starting to get their color. Here in the maritime Northwest, I do not dig up the bulbs each year, but let them stay in the ground. I have some that have lasted for 12 years, and others I have replaced over the years. One reason why some tulips die out also makes them very interesting looking in their last season or so, and he puts a link to tulip-breaking virus.
Right.
Mm-hmm.
This virus is apparently responsible for the insanity surrounding tulips in Holland during the 17th century. Though now there are varieties that are bred to be like that, which is why in my first year of growing tulips in my present garden, I did not know if the yellow and red tulip in the attached photo was already infected or just a bulb mix-up in my order of over 100 bulbs. And he sent a picture of his tulips here. Let's see if you can see that, Dickson. The red tulips, and then there's one red and yellow.
I've seen them.
And that is caused by a virus.
They're beautiful. Well, a lot of leaf variegations are also caused by viruses.
Viruses can make you better,
They can.
or they can be good.
They can make you more beautiful.
They can make you money.
Yes.
That's right. They can make you money. That's
And
right.
did he mention name of the virus, like potty virus?
It's a
It is.
potivirus, yes.
Poti?
It's a potty.
Called tulip-breaking virus.
Tulip breaking.
Yeah.
The potivirus, which is a plus-strand, single-stranded RNA virus.
Yep.
Plants with it's a flexible rod. Very interesting looking.
It's a picornavirus like.
And so he continues, if you have time this spring or next year, it would be an interesting springtime TWiV subject.
Yes.
I think he sent it last winter, so we'll do it next spring. But I think that would be interesting to get someone on to talk about that.
Well, I've suggested us having a big show on plant diseases that are viral caused, like rice blast, for instance, and that
I
sort
agree.
of thing.
I don't pay attention to what you suggest.
I do know that. It's okay. I keep suggesting, Vince. You can't
but that is a
discourage me.
joke, you know.
I
I can't tell with you. All right, have you seen the tweets from the Pathogen Posse? pathogenposse.tumblr.com. I don't tweet, but they seem to have picked up a hint from the Bronx Zoo python, and I read about them here. So, Alan, you know about the Pathogen Posse? It's a guy.
Yeah, I've been in touch with Rubiola and some of the others.
Rubiola?
Yeah, these are people on Twitter who have taken the names of viruses.
How funny.
So
Yeah, there's Rubiola. There are a couple of others now. There's a whole posse of them.
That's very funny.
Pertussis is sending out tweets, and
That's bacteria.
Yes,
Sorry.
virus and bacteria who
Sorry.
cause disease.
but yeah,
And
Come
they're
on,
all
Trichinella. I knew Trichinella very well, as a matter of fact.
They are. The point of it is to be proponents of immunization.
yeah,
Right.
yeah. They're all most of them. Well, norovirus is also sending out tweets, but most of them are vaccine preventable diseases that are tweeting.
The Pathogen Posse. That's good.
Yes.
It's good.
Yeah.
It's a good cause. All right. Thank you, Chris. The next one is from Tim. I enjoyed listening to your latest podcast, TWiV 128, with the exception of Alan's horrendous comment about eating endangered mountain gorillas.
What?
He said he liked them fried instead of boiled or
Oh,
something.
my God.
He was joking.
Or gorilla.
Hey, my daughter likes Gorilla Munch.
Really?
It's a cereal, Dickson.
It's a cereal.
Oh, okay.
Hey, you know about that too?
Yeah, my daughter.
Stop, all of you.
All right. I'm sorry that he offended you. I also especially enjoyed your combination of co-hosts. One thing that I like about TWiV is that you take the time sometimes to research the subjects and the subject posed by a reader. I think that's so funny. Sometimes,
Okay. Guilty as charged.
All right. We'll tell you the system. It's every third show we research.
I guess I still like to see people do their homework. Since I just dissed Alan, I want to know that it's obvious that Alan does his homework every week and the show benefits. All right.
There you go. All right.
It does.
The question has been bugging me for some time, and having recently viewed the NIH conference on State of Knowledge workshop, myalgic encephalomyelitis, chronic fatigue syndrome research, and after watching the antagonistic exchange between Dr. Coffin and Dr. Mikovits, I was hoping you might discuss this subject. So he sends a link to the YouTube video, and he says, does the federal government or the institutions that employ virologists and other highly specialized scientists dependent on outside funding ever get training in interpersonal communication, conflict resolution skills?
Never.
No.
No.
No, it's 30 paces and pistols.
If not, why not? The apparent lack of prior communication between two of the most important scientists involved in XMRV research baffles me, especially since at least one of them is spending taxpayer dollars. However, I want to stay out of the XMRV realm here. It just serves as a good example of this type of lack of communication conflict, and it just happened to be very public. The history of science is dotted with egotistical conflicts among noted scientists, many times to the detriment of the process. I suspect it's partially due to the fact that everybody is often scrambling for funding from the same pot of money and something else. I have worked for many large organizations, but none so large as the federal or a state government, and most of them have always provided training on communication skills, which helps even the reluctant pupils. As an added note, I have a background as a scientist. Thanks for putting the time you do into this program. Tim is from Colorado.
Yeah, Tim, I must tell you that there are two kinds of people out there, those that like to talk and communicate and those that don't. And the ones that do end up as either professors and teaching or in doing some kind of instructional work. And you're listening to a podcast that is filled with those kind of people. I mean, we have a passion for communicating. So we're more than willing to sit down with anybody and listen to their ideas. And then, using the scientific method as a basis for criticizing, go ahead and tear the idea apart and see how well it holds up to analysis that's critical and objective. It's hard to find people that are working on the same thing and coming up with two different results to go to a meeting and to be objective about their findings. I've found this is true no matter what, whether it's due to the cause of drug-resistant malaria, for instance, was a good example in my day of two groups that were working separately. One was at NIH, one was at Harvard. They both had a similar result, but they found a different genetic basis for it. And it turns out that they were both right. And they argued and fought and wasted a lot of energy with just accusing each other of not doing the right thing. And they both did the right thing, and they both found the same cause. They didn't find the same cause. They found two different causes for drug-resistant malaria. And they never expected there to be two causes. Well, there might be 5 causes or 20. Who cares? I mean, the point is, if you remain objective about science, then you shouldn't take out your passion for science on another scientist.
But this is an inherent occupational risk.
It's people.
It's people, right? It's people doing science, and you're finding out information in incomplete fits and starts, and you get a partial data set.
Yeah.
And of course, the person who's put in all this time to get these data believes them to be true, and somebody else puts in all their time to get data on something similar and finds divergent results. And it can get tense from time to time.
Well, they just had Johansson and Leakey at the Museum of Natural History sit down and resolve their lifelong differences that they've had all these years. I mean, from Lucy to all these other anthropological findings that show how complicated our origins really are. In those days, it wasn't complicated. These two guys thought they had the answer, and they didn't, of course.
No, this is pervasive in science. I mean, in virology, the most famous conflict was between Albert Sabin and Jonas Salk.
Hear, hear.
Publicly, criticizing one another. And I think you could give them all the conflict what is it called?
Conflict resolution training. Yeah.
And then you get up there in front of an audience, and some guy says something, you're wrong, and the other guy's going, I am not wrong.
It's a fist fight.
Yeah.
Right. And also, the personalities who are drawn to science are people who don't take things at face value.
Yeah.
If you're the kind of person who's agreeable and goes along and says, "Oh, well, I'll just let them say that," then you're probably not in science. We're the ones who probably sat in the back of the classroom telling the teacher they were wrong when we were in elementary school. This is who goes into looking at the natural world to get at the fundamental data.
I looked at this YouTube video that he gave us, and in fact, it's a pretty minor dust-up. I've seen a lot worse.
Yes, absolutely.
Yeah.
So I feel that it's not viewed as objectionable, say, by the NIH, because, when fraud became a big issue a number of years ago, now all our students have to take fraud courses on fraud in science.
That's right. Ethics. We do take ethics.
And this is mandated by the NIH, but they don't mandate that we take conflict resolution, nor communication skills, and that will never happen.
Yeah, and in the one in the YouTube video, nothing got broken, nothing got thrown. So I've
no
seen
harm, no foul.
Yeah.
Personally, I don't do that sort of thing.
I understand.
Actually, I did it once.
Did you?
Yeah.
Did you?
I agree with him that it's a waste. It's a waste of time and energy.
It is.
It does get in the way of progress. Egos get in the way of progress. It makes much more sense if people can sort of work this stuff out in a calm, collected fashion. But as we've already said, the personalities are not always compatible with
that. You should go into an operating theater, and then you'll see personalities.
Well, there's a side effect to this criticism also, and that is that every time a study section meets, and you sit there and you don't know anything about this subject, and you get two of these reviewers at opposite ends of the spectrum of whether this guy is deserving of his money or not. It just drove me crazy. Or her. I'm sorry.
Amit, you seem like a calm guy. Do you ever have conflicts with other scientists?
Not till now.
Now meaning...
Not until... You mean this moment? Or?
Not until now, but, I'm listening to this.
So in India, where you were trained, is the culture just like science everywhere else? People get in fights, verbal fights, or not?
Yes, they were getting in fights, and I think the case we are discussing about this, these two people coughing, and they are not ready to meet in personal, so they met in public, and then the anger poured there. In India, I think we can also have these fights in personal setting or maybe in public.
It's international.
Yeah, it's international.
All right, let's see here what we have. Maybe one more. This is from Kevin. Have you guys seen these gorgeous microscopic photos of various drinks?
Drinks?
Drinks.
Yes.
A microscopy of drinks?
Yeah, cocktails through the
you gotta
microscope.
have a look at those. They're really pretty.
Yeah, cocktails, passion fruit martini, Bloody Mary, pina colada. They have pictures of the microscopic pictures. Very pretty.
These things look like geological samples.
Shirley Temple. Look, they have them labeled. Shirley Temple. Pina colada.
What do they do? Dry them down and look at the crystals?
Sake. Let's see here. It's just close up. Is it dried down?
I'm pretty sure they're dried down.
Right.
Because these are definitely crystal shots.
Yeah, they are crystals. They must be dried down. So this guy,
But most of these drinks are best served in crystal anyway.
Indeed.
He says, I think I'm going to buy some tequila. There's actually a page of, here's tequila right here, dried down.
Yeah.
Crystals
that's
of tequila.
nice.
Very, very modernist.
Very nice.
Indeed.
You can even buy a poster of it if you'd like and put it on your wall. Are there any interest, to keep this on topic for TWiV and TWIP, what kind of microorganisms threaten our beloved agave population? I couldn't find any. I looked for viruses of agave, but I didn't find any. So you're okay. What is agave used for? Tequila?
That's tequila.
They must have some insects that like to eat them, though. All plants have insect pests.
I looked for viruses of agave. I didn't find any.
By the way, you know that almost half of the plant viruses that get into a plant get there through the bite of an insect.
Yes, yeah.
Because the plant is pretty resistant to the outside.
Here's one for
Amit,
you.
there's your next project.
Yeah.
Agave viruses.
Yes, once I will be done with this one.
He's got his hands full.
Exactly.
Dickson, this one's for you. Are there any interesting immune system tricks that flowering plants use to encourage pollination but restrict parasitism?
Eek. That's not for me. I'm not an expert at that. I'm sorry. I will deflect that question off to a plant biologist. I'm not a plant biologist. I don't know. The answer is I don't know, but that's a great question.
None of us know.
It's a good question.
Well, maybe there's a lister. Okay,
right, let us know.
Yeah, there's a virus of prickly pear cactus that I've found, but not finding. And agaves do have various pathological problems, bacterial and such.
Okay.
The one thing I know about flowering plants is that they emit a spectrum of reflection that's outside our range of detection, but that insects can actually pick it up. So they look like almost like an ultraviolet spectrum that the insect can actually see. Not all of them, only the ones that are involved in the pollination process. So it's really quite interesting how insects and plants have evolved to sort of end up with a mutualistic behavior. So that's the best I can do on that question. Sorry.
Thank you, Kevin. Kevin is from Santa Barbara.
Okay.
Nice place. All right.
It is a good place.
Let us do some picks of the week.
Yes.
You're so excited. Let's start with you.
I am, because I read this article. And
you don't read the other picks, huh?
Well, some I do and some I don't understand. I read them and I don't understand them. But this one is pretty interesting from several standpoints, and you guys can add to it, and I'm sure you're aware of this one. It's called Human Cytomegalovirus Directly Induces the Antiviral Protein Viperin to Enhance Infectivity. This was in the latest issue of Science. And it says, in effect, that although viperin is a protein that's elicited by interferon production, and it's supposed to interfere with viral replication, in this particular case, the cytomegalovirus actually benefits from the induction of viperin, because when you knock it out, the virus doesn't succeed. And this is a normal pathway for antiviral protection in a host cell that this particular virus has taken advantage of to actually get around and actually be able to replicate inside of a cell that ordinarily would kill you off. And the protein viperin, it translocates from the endoplasmic reticulum to the mitochondria. And how that actually ends up interfering with viral replication, I don't know, but you guys know more about that than I do. But I just found it interesting that here was yet another trick that the viruses have learned by taking advantage of our cellular biology that's absolutely unaltered.
Yeah, that's very interesting, because viperin is induced by interferon. It is presumed to be antiviral.
That's what it says.
And
but
cytomegalovirus has evolved
exactly right.
to need it.
And this paper throws up its hands and says, we don't understand what's going on here because this shouldn't work, and yet it does. They've got the proof.
It's really cool.
Yeah, I think it's
Maybe
cool.
this one ought to go on the list, huh?
Hey.
Yeah, I think so.
Put that one on the list.
We'll put this on the TWiV list, which is now,
there you
like
go.
a page long.
Okay.
And it's got some pretty pictures in there also.
Rich, what do you have?
I have a story that could go under the heading NIH Rocket Boys. Okay, so there's an NIH site that I put the link in here too that summarizes this whole thing. And the gist of this is that in 1957, a young boy named Terrence Boylan, who at the time was 9 years old, he and his buddy were trying to build rockets. And they were having various difficulties, one of which was the reagents were getting expensive, right? They were running out of money.
Right.
So Terrence's father, in fact, turns out was a physician scientist at State University of New York at Buffalo. These guys lived in Snyder, which is a suburb of Buffalo. And so Terrence went in and asked his dad, this is 1957, he asked his dad, Dad, where do you get your money to do your research? And his dad, sort of half listening to him, says, I get it from NIH. And so Terrence wrote a letter to the NIH that says, Dear Sir, my friend and I are very interested in space travel and have a great idea for a rocket ship. We were wondering if we could have a little sum of money, $10, to fulfill our project. We would be most grateful if you would send it to us. And he signs it off. And this came to the attention of Ernest Allen, who basically was the guy who got, if I understand it correctly, basically spearheaded getting the NIH extramural program off the ground and organized. Wow. Okay? And Allen took an interest in this. And It's interesting because these guys basically got triaged.
Right.
But he actually took it to a review committee. This is a great letter that came back to him. It says, "Dear Terrence, your request for $10 in support of your rocket ship project was read to the National Health Advisory Council on March 12th. This is his summary statement, okay?
All right.
The members of the council agreed that your proposal does not fall in the health and related sciences fields for which they recommend research support. On the other hand, they agreed enthusiastically that you should receive the funds requested and contributed as individuals to make the support possible. In accordance with the wishes of the council, I am therefore pleased to forward my check in the amount of $10.
Outstanding.
Please accept our very best wishes for a successful project." These guys used the money. They built a rocket that ultimately they shot it up and it went out of sight. And somebody picked it up in Clarence, a neighboring suburb. I guess it had some sort of payload that identified where it came from. So the money was well used.
Spent, as it were.
Terrence since has become a musician. Of some repute, apparently, and but still promotes science education and et cetera when he has the opportunity. And this whole thing has spawned a whole bunch of educational activities within the NIH for kids. That's all pointed to on this site. So I thought it was a great story, nice history. There's a whole link here, the whole page here at the Center for Scientific Review that has a bunch of different links to this story and the stuff that has spun off from it. Funny. So I thought people might be interested.
Beautiful.
That's great.
It's very good.
Alan, what do you have?
Well, since you stole my pick, Vincent.
Did I really?
No, Vincent, not Dickson.
Vince.
Our names sound similar.
Sorry.
We'll get to that in a moment. But I have a YouTube video. Since we're on the kids in science theme, and this one I saw a few weeks ago. It's a video from I guess it's from NOAA, National Oceanic Atmospheric Administration, not the guy who supposedly built an ark.
No. To go in the ocean.
Yes, to go in the ocean. So it's this video called "What Do Marine Mammals Eat?" And it's they did this project with elementary age kids. And a scientist, a researcher who studies exactly this problem. And first they start off with asking the kids, how would you figure out what a marine mammal eats, what a seal or a dolphin eats? And the kids start throwing out ideas. Well, you could try and you could put a camera on its head. You could try and try different baits with it. And the researcher, they cut to him and he explains, that's an interesting approach, but here are the difficulties with it. And finally, one of the kids says, well, I guess you could look at their poop and figure out what they ate. And the researcher says, exactly.
Ta-da.
And then he goes through the procedure that they use to determine what marine mammals eat. And it's just a neat little snapshot of the scientific process. You think about how to do the experiment, then you do the experiment and you get the answer. And it's a really nice video.
Neat.
Nice.
Very cool. I'm looking at it here. It looks really cool.
Yeah.
Amit, did you want to pick something?
No, actually, I didn't.
Okay. Very good.
I didn't know that.
It's okay.
Yeah.
I picked something, and of course, I guess I stole it from Alan, although I picked it not knowing that this would be his. But not too long ago, the National Academy of Sciences Press announced that all of its PDFs would be free. All PDF books free to download as of June 2nd. And we have, I believe, selected some of these books as picks before on TWiV. And some of them cost money, even the PDFs. But now they're all free, which is great. It's good to see the government jumping onto open access.
Yeah, and it used to be, they had an arrangement on their website for a long time where these books were frequently put out where you would have to pay for the print version, or you could read the PDF version online, but you couldn't download it.
You couldn't own it. You couldn't own it.
So now you can download it. Yeah.
Have it. Now you can download it. And in fact, I can probably go ahead and disclose at this point, I needed one of the reports once, and I actually wrote a little script that did a web page scraping in order to effectively
Good.
PDF. And while I was doing it, it took me about an hour to write this thing, and then scrape the PDF, and then get the downloaded report so I could read it offline. And I thought, this is ridiculous. This is a government-funded study that really ought to be free.
No, this is totally appropriate. The government should not be charging.
And actually, Alan, maybe you can comment on this, but my sense is that the government has really been pretty forward in this open access stuff. They've driven this stuff of, any government-funded paper has to be open access within at least a certain period of time after it's published.
Yes, in the past few years especially, and it's ramped up recently that I've seen, because I encounter this a lot as a reporter. Of course, as a reporter, I can just send an email to the researchers and say, hey, can you send me a copy of the paper? And that always works. But it's a little bit of an annoyance if I point to something, especially if it's, for example, on my blog, and I want to link to something, but I know people can't actually read it unless they pay some exorbitant fee. And I certainly sympathize with publishers who help pay my mortgage, but there's a lot that could be done with open access, and NIH and other government organizations have been kind of pushing that, and in the past couple of years they've accelerated it as the technology has gotten better, and there's this real. Yeah. I think there's now a real. They kind of got religion on this, and they're serious about making things accessible to the people who paid for them.
Mm-hmm. Although the National Academy, the proceedings of the National Academy of Sciences is not free unless the authors pay to make it accessible, and I don't think that's appropriate for a government publication.
Well, now wait a second here. There's an economic problem here.
What's the problem?
The problem is it costs money to put out a journal.
You have to print it,
and it costs a lot, even if you don't print it. Everybody in the open access community likes to say, well, peer review is free, therefore it shouldn't cost anything to publish a journal. And that completely misses that somebody has to receive all these papers, triage them, decide which ones need to be reviewed in the first place, then pick the peer reviewers and send them out. That entire process requires somebody. You can't just hand that off to a $10 an hour intern. You need somebody who knows the science that they're reviewing, and there are research editors at every journal who handle that. And these people are not cheap.
But that's what page charges are for.
Exactly, and that's what you're objecting to.
No, I'm objecting to the $1,500 charge if you want your article accessible immediately online, as opposed to a few. But that's not page charges. It's in addition to the page charges.
Yes, the page charges were to defray some of their expenses. The open access publishing model is based on page charges.
Right, so PLOS charges a page charge, and that's used, and then everyone gets access. But PNAS charges a page charge, but you don't get open access unless you pay another $1,500. That's just
and that's because they can't afford currently to do it any other way. I mean, I would love to see them get more money, but that doesn't seem to be in the cards these days.
No.
Well, part of the problem is all this process that you mentioned, and maybe it could be streamlined. These are nicely set in type into PDFs, but maybe that's not. PLOS doesn't do that. They just put it online as a HTML document, not a PDF. So actually, that's not true. They do put PDFs.
They do put
PDFs, but there's a whole other issue that happens when you're not releasing a print journal. Which I've been talking about for years, and nobody seems to be taking very seriously, in my opinion, which is archiving. I've got computer disks from 5 years ago that I can't read.
Right.
We are now taking the entire scientific heritage of mankind and putting it into a digital format, which we don't know if anybody's going to be able to read this in the future.
Yeah, you're right.
So if you don't have a print journal, everybody likes to pick on how old school print is, but it has one overwhelming advantage, which is that you can pick up 1,000-year-old text, and with absolutely no hardware, you can read it.
That's correct.
It's true.
Got it right.
So that's my little shtick on that.
Yeah. So let me just summarize here. We've all made picks, and I'm the only non-virologist here. And I picked a virology paper, and all the other virologists picked non-virology picks. Now, this is a kind of a reversal of fortune here.
We go back and forth, Dickson.
I love it. I know. I just like the feeling of being the one to pick
you're not
the
the
virus
only one.
paper.
There's 2 picks, but there are 2 listener picks of the week.
Yes.
Okay, 2 students from my virology class.
I didn't know that. Okay.
Sent articles about the man who received a stem cell transplant and was cured of AIDS.
Yeah, I remember that one.
So we talked about this a long time ago, and there's some follow-up articles in a San Francisco.
He had leukemia, as I recall. Yes.
CBS San Francisco and the New York Times, which describe his life at the moment.
Right.
So that's
look like nice articles.
Very nice. Yep. And thanks to Adriana and Ye Jung, who were students in my class. Hope you are still listening, because otherwise I'd take your grade back.
We can change these things.
I can't do that. Just kidding. Well, they sent it to me just a week or 2 ago, so they're still looking at virology.
This matches well with that other TWiV that we had on that subject, where you had long-term T-cell storage of viruses in the bone marrow. Yeah. So that's cool stuff.
Okay, that's TWiV number 137, and I want to thank Ian Lipkin at the Center for Infection and Immunity for being here. We'll have him back again. Amit Kapoor is also at the Center for Infection and Immunity. Thanks so much for joining us, Amit.
Thank you for inviting me.
Great work. Looking forward to more of it.
Thank you.
Really nice. Alan Dove. Alan Dove is at alandove.com.
Always a pleasure.
Hope you recover from your lightning strikes.
Yes, it was rather shocking at the time, but I'll probably get past it.
Rich Condit is usually in Gainesville, Florida, but today is in Texas. Good luck with your grandchild.
Yeah, absolutely.
Thanks very much.
Yeah.
And Dickson Despommier is who knows where.
I'm right here.
At verticalfarm.com. I'm Vincent Racaniello, and I'm at virology.ws. You can find TWiV on iTunes, at the Zune Marketplace, or at twiv.tv. If you have a smartphone, an iPhone, or an Android device, you can get an app to stream the episodes to that device. You can find that at microbeworld.org/app. Send us your questions and comments to TWiV at twiv.tv. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.